So we are coming full circle back to chemistry. So we have Gabriela Schlau-Cohen, who is a member of the chemistry department here, and is going to talk about sunburn.
Or rather, I'm going to talk about the lack thereof. So unlike you and me, these plants here can enjoy this beautiful sunny day without slathering on SPF 50. And so I'm going to talk to you about developing sensitive detection approaches that allow us to understand the underlying molecular mechanisms behind plants' version of sunscreen and how we can use that to address food challenges. So this is my version of the slide that everybody's been showing this session about how by 2050 we need to double agricultural output. And this is even more challenging because agricultural output is actually expected to decrease by 25% due to the effects of climate change. So this is going to lead to a mismatch between the projected food demand, shown here, and that projected agricultural output, shown here, for a region in sub-Saharan Africa, where this mismatch is particularly striking. So we need new technologies and new approaches to bridge this gap. And we want to think about ways that we can begin to potentially increase this projected agricultural output. So if we think about crops, one place where crops are lost is in the very early steps of photosynthesis. So in the first steps of photosynthesis, under some conditions, like the sunny day you see here, up to 70% of the absorbed energy is actually converted to heat and lost into the environment. So we want to think about ways we can actually recover some of that heat and use it to drive crop production. So the reason for this loss of energy basically comes from the fact that the intensity of sunlight changes. It changes from day to night, and it changes as clouds pass over. So this changing in sunlight actually poses a challenge for a plant. And that challenge is also shown right here. So this is sunlight on a typical day and a typical point on a crop field. We see sunrise, sunset, clouds, and shadows. Now for a plant, sunlight up until around this level is useful. This is the energy that matches the capacity of the molecular machinery of a plant. So that's used to drive photosynthesis and crop production. Energy in excess of this is dangerous. That energy exceeds the capacity of the molecular machinery and can cause the generation of damaging products. So what plants have done is they've actually evolved a regulatory cycle. This is their version of sunscreen, where they take this input and they want to keep the useful and only the useful energy. So this regulatory cycle actually is implemented really cautiously. Plants, instead of slathering on that SPF 50, they slather on SPF 500. So they dissipate even more energy than they need to. And so we want to think about, can we optimize this regulatory process to increase crop yields? So what's known is this regulation is actually implemented through a molecular level switch. Under high light or sunny conditions, this switch gets turned on and the energy is dissipated as heat. Under cloudy conditions or low light conditions, that switch gets turned off and the energy is used to drive productive photosynthesis. So one of the things that we study in my lab is what is this switch and what are the dynamics by which it's toggled on and off. So what's known, at least in green algae and moss, which are good model systems to study this process, this switch is this protein right here. It's a protein called light harvesting complex stress related, or LHCSR, and it's a chlorophyll-containing protein. Now, if we want to sense the action of this switch, what we do is we use fluorescence. And that's because when the chlorophyll within this protein absorbs sunlight, in the case of that switch being turned on, that sunlight gets converted to heat. If that switch is turned off, that absorbed sunlight comes back out as fluorescence. And so that's why we need sensitive and robust ways to detect fluorescence to characterize the state of this switch. And what's emerged in recent years are really exciting new ways to detect fluorescence from photosynthetic systems on extremely different length scales. So there's actually become an ability to detect photosynthetic fluorescence on the planetary scale. So what you can see is this is data from satellites. And if I can find my mouse. And what you'll see in a second is switching to red. We're actually looking at the fluorescence from the chlorophyll. We can see it grow in the summer, decrease in the winter, and then grow back in again in the summer. And so satellites are able to detect chlorophyll fluorescence with a spatial resolution of around 10 miles. And by correlating the chlorophyll fluorescence with different local weather conditions, we can get an understanding of the large-scale behaviors or the responses of crops to these changes in weather. So where crops are working optimally and where they're not. And what we do in my lab is we think about that and want to zoom in on the molecular mechanisms behind these types of chlorophyll fluorescence. So we go to the extreme opposite length scales. Instead of looking at 10 miles, we look at a few nanometers. And so we do that by detecting fluorescence from individual proteins. We take an extremely sensitive microscope, a modified confocal microscope, that has the detection ability to detect fluorescence from individual proteins. And so we monitor that fluorescence for one protein, and then for many, one protein at a time, building up statistics for many, many different proteins. And we get this distribution of behaviors. And so what we can see is that there are immediately two populations which tell us about two different conformations of the protein. So here we have brighter fluorescence, which tells us that the switch is off. Here we have less fluorescence, which tells us that the switch is turned on. And this experiment was done under conditions that mimic a slow change in sunlight, something like a sunrise. And so we were able to characterize the switch on and switch off, as well as the dynamics by which the protein turns that switch on and off under these conditions. And we also looked at it under conditions of a fast change, something like a cloud passing over. And what we saw was a new conformation here, which tells us about there's actually a second way to turn the switch on. So overall, what this is showing us is that what plants are doing is they're able to turn on their sunscreen in two different ways. They turn it on in one way in response to a sudden change. And from the experiments I showed you, as well as many others, we were able to identify a molecular level picture of how that's done, that it's a rigidification of that protein structure. We also saw that that switch gets turned on in a different way, a sort of slower, more gradual way, and that's done by actually changing the molecular composition. And so the reason for this, like I said before, is that if we think about how sunlight is changing on a typical day in a typical crop field, we see these slow changes from sunrise, as well as these fast changes from clouds passing over. So plants have developed not just one type of sunscreen, but actually two types that respond to the two types of changes in solar intensity. So this is a picture of how the world works. But if we want to improve our agriculture, we have to improve this sunscreen. And so one way we can think about doing that is by knocking out this gradual change. Having this fast change will still provide us some protection, potentially be good enough. And by knocking out that gradual change, we can imagine getting closer to this picture here. So in this way, we can use our understanding of the mechanisms of regulation and the discovery that we did in my lab that there are actually two types of sunscreen to remove an unnecessary, perhaps overly cautious type, to get this increase in crop yields up to 30% according to our simulations to meet the need for increased agricultural output. So I have to end thanking the people that actually did this work, particularly a postdoc, Toru Kondo in my lab, and a graduate student, Weijia Chen. And I'm happy to answer questions along with the rest of the panel.
Okay. Thank you. So yeah, so we have our speakers come up and we have some seats here. Really diverse range of topics here, but one thing that was common is electromagnetism was in all of them, right? Anyway, open to the audience here for any questions.
Yes, I'll move forward.
Hi, my question is for the first panelist. You worked with the DJI drones. The DJI drones.
Yes, we did.
I'm wondering how did you work around the FAA requirements? You know, there has to be line of sight at all times.
Yeah, so I guess when we did the experiments, we were mostly, like we had a person who actually programmed the drones and made them fly, so they were mostly line of sight all the time.
Okay.
Yeah, so that's how we do it.
Thank you.
Other questions? Yeah, one. Oh, go ahead. Yeah.
Yeah, so hopefully everyone can hear me. So Deepak, very quick question. It's very interesting with the mapping and with the drones, et cetera. Is there some plan that this would migrate into some automation for administration of, watering, irrigation, or chemical applications through some automated process?
So our current focus is on doing it for agriculture, right? So for agriculture, we are actually doing a lot of collaborations with companies and with farmers themselves on irrigation systems and on pH. I think these are the things that we currently work on. But we are actively looking for other things to sense in general. We have plans to use RF as a sensing modality in itself, and as well as look for other sensors that we can use to interpolate these things. The other kind of thing that we do is to use, the drones that we use so far have RGB cameras, and we want to replace them with multispectral cameras and see what extra things can we sense based from the drones themselves. And does that answer your question? Thanks, yeah.
So I have a question for Gabriela. How easy do you think it will be to mutate or modify, or maybe through breeding, or I don't know exactly, plants? Do you have some speculation on that?
Yeah, so it's actually really easy. The first step of it was already done, where everything involved with plant sunscreen was just upregulated, and that got about a 20% increase in crop yields. It was the cover of Science and in the New York Times. And so what our work is doing is saying, all right, let's not just blindly upregulate, let's upregulate with some sort of understanding of what each part of this process is doing. And so that will allow us to get from the 20% that was done there to 30 or even a little bit more.
Was there a particular crop that was done with? Or?
That was done with corn.
Corn, okay.
Yeah, maize.
Corn's big.
UISC.
Just to follow up on that, why didn't evolution take care of that?
Evolution has different goals than we do. The plant wants to survive and reproduce. We want crops.
One way back there.
Thank you. So I have another plant-related question. What is the relationship between the switch you talked about and the switch that cause some plants like algae to produce like additional lipids or starch in response to photooxidative stress? Do those switches interact, or?
I don't think it's totally clear how they interact, but they're sort of compartmentalized by the organization of the algae. So this switch, in oxygenic photosynthesis, it drives water splitting, which causes a proton accumulation from the splitting. So this switch is regulated by pH. So as the pH drops, it turns the switch on and off. Whereas that oxidative stress, so this can prevent oxidative stress, but then this oxidative stress, my understanding is that's a genetic regulatory pathway sensitized by singlet oxygen. So it's sort of like the byproduct of a mismatch of an error in this switch can activate that one.
Oh, so this switch doesn't require anything to be upregulated or downregulated. It's all within the protein?
It's all within the membrane, because this is a membrane-bound protein, so on one side you have a pH drop, which triggers some enzymes as well as acts directly on that protein. So there's no genetic component to this piece of it. It's a shorter timescale process.
So I have a question for Maren, and forgive me if I didn't get all the physics right, or follow it completely, but with your X-ray sources, can they be coherent? Can you have pulsed and either very short time frames that can allow new types of structural spectroscopy, or what are your kind of intended, where do you think the bounds of where this can go are?
Yeah, so that's a great question. Yeah, so we are currently exploring the answers to all of your questions. So in principle, you could do all of the things that you said, and currently we are quite a bit focusing on seeing how well you could do in terms of making them coherent, in terms of spatial coherency, and then also temporal coherency. So those things are possible. I cannot tell you yet how well exactly that will work. So that's kind of what we're focusing on that right now, but it's kind of work in progress.
But what about kind of time frame? Could you get something pulsed? I mean, the dream is to watch molecules in real time, right, through X-rays scattering. Is that something you think this can do?
What wavelengths are of particular interest to you?
Usually angstrom and a half or to 2, maybe, usually for chemical structures.
Right, yeah. So those are the type of things that we are currently exploring. So, but I would say it depends on the effort put into this, but I think of the order of, if it's doable, it should be doable within, let's call it a few years. It is very unpredictable in research and science, but the fact that we're working with this like compact sources and so on, that makes the whole thing much more controllable, and we can make progress faster than if we had to rely on, like very large sources and so on. So that's why I'm a bit bullish about this, right?
Well, that's fascinating. Okay.
One more back here.
Okay.
Back to the sunscreen for a second. You said the switch was driven by a pH change. What causes the pH change?
So when there's sunlight, high-level sunlight drives a water splitting, which causes the generation of oxygen and protons. Those protons power the ATPase, which causes ATP. So if there's too many protons and the ATPase can keep up with them, those protons just accumulate within the leaf. And so as those protons accumulate, that's in effect the drop in pH.
Other questions? Right there.
A question also related to pH for Deepak. Deepak, you seem to have some way of measuring pH using the drones. Could you explain a little bit about how that's done?
Yeah, so essentially what I did not talk about a lot was what is the machine learning algorithm that exactly runs on top of things. But for the pH map that we generated, what we did was we did a sparse sampling of pH on the field itself using sensors. And we trained a model that could map visual features to pH values based on those samples. And then we extrapolated or like predicted on the rest of the field essentially. That's what we do. So we use this machine learning tool called Gaussian processes, which can be trained using a small number of samples if you're familiar with that. But I'm happy to like talk more offline.
Yeah. So one of the things I was wondering about is these switches, you know, with the drone, you know, whether it's fluorescence or whatever, you have pH switches, stress to the plants. Is that something that you think has some viability in terms of looking at native fluorescence? I don't know what kind of wavelengths, even if there's variation, if that would be a way to look at precision agriculture.
Yeah, that's actually being done with the satellites, so presumably it could be done with drones too. In plants, there are two photosystems, photosystem 1 and 2, which fluoresce at slightly different wavelengths. So by looking at the ratio of the two, you can also get a sense of those things, which is basically 700 nanometers versus 680 nanometers. So presumably it could also be done with drones because it's being done with satellites.
Could you actively excite too and learn something by doing an active excitation and maybe doing time resolution on a drone? Or is that too much?
Yeah, so there are, in terms of, I think there was a discussion earlier about miniaturization of devices. So one area that's really been increasing within the plant community is devices where you can go out in the field and measure actually all of those things in the field. I don't know if people have gotten it in the drone, but you could imagine, I mean, using a laser, you know, it deflects the same way.
Flying around
like,
at night, for example.
Yeah, or like a guide star, right?
Yeah, okay.
Yeah, time is.
A new hazard.
Can you envision your drone can actually pick up herbicides and pesticides?
Yeah, that's an active question for us. It's hard to say anything conclusively. Yeah, like we would want to do that, but we don't know. We're still looking into it. Yeah.
I have a follow-up question.
Sure.
Do you foresee rolling out this technology in any kind of scale being difficult until the FAA changes some regulatory models where drones can be allowed to fly autonomously?
Yeah, so in some ways we're looking at this in a global perspective. So there are these regulations which are like restricting us in the developing world, for example. Like we deployed this in India and Africa, and there like flying drones is not as easy as it's in the US. And what we've done there is to come up with alternative solutions, like using helium balloons with smartphones, because like drones are expensive for those cases too. Like in India, for example, $1,000 is a lot, and in Africa, $1,000 is a lot. Like So what we end up doing is that we use helium balloons with smartphones, because people have smartphones in India these days, and also in Africa, the smartphone penetration keeps going up. And more often than not, like these cheap smartphones have good cameras. So what we end up doing is that we send these cameras up using helium balloons, and it's easy to get labor to walk around, and there's someone who walks around the field, and it's like a semi-drone kind of solution. So we come up with ways to like get around regulation more often than not. But that said, like definitely we would want regulation to be easier on flying drones and collecting this data.
Yes, Gabriela, have you ever applied your research in a controlled environment that uses supplemental grow lights? And if so, do you think it has any impact on your work?
So these processes have been studied in, we can imagine, 3 conditions. So my particular focus within this scope is on isolated proteins, understanding the underlying molecular mechanisms. Then there are people who use growth chambers, kind of go from 1,000 microeinsteins to a couple hundred. And then there's field studies. And in terms of understanding the organism-wide behavior, going to the field studies has been critically important in recent years, like particularly this science paper that I mentioned before. Because in the growth chambers, there's this variation, you know, you can go low light, high light, but as I showed in the plot, there's a really much more dynamic profile of sunlight that's so difficult to mimic. So I think that's really important.
Gabriela, you had mentioned that you can envision humans using this switch on, switch off mechanism. So how can that be applied to humans for the sunscreen? Had you or maybe someone else did? I'm sorry. Is it possible to use this idea for human sunscreen?
If I said that, I'm sorry.
I'm sorry.
No,
Maybe someone else.
I mean, one could imagine some sort of photo-switchable polymer, right? So that I think one of the challenges with sunscreens, like chemical blockers, is they're always active. There are a lot of byproducts that cause cancer even in the dark, right? They're redox active in the dark. And so one wants a better sunscreen in order to enable that. And that's where one could imagine turning to my synthetic colleagues to develop some sort of photoactive sunscreen that's more passive.
Great. Okay, well, I think it's time for a little networking, and we'll reconvene here in about 15, 20 minutes.
Thank you.